But yeah, I'm excited to kick off the conference this year. So it's been a long year, 2025. Lots of advances. There's been interesting trials at ASCO and AACR, and most recently ESMO in Berlin had some interesting new studies too. And I guess most exciting from our point of view, from Katie's and my point of view, is that we have two new colleagues, Dr. Afsaneh Amouzegar and Dr. Derrick Tao, who joined us from MD Anderson, and actually both from MD Anderson. Uh, so, uh, and they're interested in different aspects of melanoma. Dr. Tao is interested in cellular therapy and is going to be heading up some clinical trials for cellular therapy at UCSF. And then Dr. Amouzegar is interested in brain uh, metastasis and CNS melanoma and other rare subtypes of melanoma. So we look forward to having them join us and talk to us. And I'll let Dr. Tsai introduce herself, who you already know, I think.
Thanks, Adil. I'm Katy Tsai, one of the medical oncologists here at UCSF. I think I may know a fair number of you already, but just wanted to introduce myself and say hello. As Adil just mentioned, we're absolutely delighted to have two new faculty join us, Dr. Derrick Tao and Dr. Afsaneh Amouzegar, who we'll get to meet later during this webinar. And we're just so excited. It's remained an exciting time for melanoma treatment, and we're so happy that you could join us today.
So maybe I should get started just in the interest of time while Dr. Muzigar and Dr. Tao uh, join us, if that's okay.
Sure. I'll do what I can. I'll see what I can do in the background, but if you want to get started, that would be perfect.
Okay, great. So let me share my slides and hopefully that's clear and everybody can see my slides. So my talk focuses on adjuvant and neoadjuvant therapy. And so these terms that we use a lot of the time are kind of confusing, and I just wanted to spend a minute to just clarify what exactly this means. So as many of you know, when you have melanoma, it's on your skin, and in our first step is to consider surgery. And that's the way it's always been treated. And usually once the dermatologist or your primary care physician or um, surgeon sees the melanoma, the first step is usually to take people to surgery and first of all confirm that it's melanoma, get uh, what we call pathologic features like the breast floor depth, like how thick the melanoma is, whether there's any invasion of underlying structures, and whether it's spread to lymph nodes. And typically, that has been the way, like in that first line there, that's the way melanoma has been treated. And we'll often sit down and talk to patients and say, okay, looks like your melanoma is at a high risk of recurrence. And so we are considering immunotherapy or considering targeted therapy, and here are the pros and cons of doing it. And what's happened in the last I want to say 5 to 10 years is this idea that you can actually give immunotherapy or targeted therapy prior to the surgery. And uh, somewhat surprisingly, it seems like one of these two approaches is clearly better. And so there was a study done in primarily in Australia and in Europe called NADINA. And basically what happened in the study is that, patients who had stage 3 melanoma, so meaning the melanoma had already spread to lymph nodes, were uh, randomized or randomly assigned to either get uh, 2 cycles of immunotherapy followed by surgery, or surgery followed by just immunotherapy. And the type of immunotherapy that was used in arm A was this ipilimumab plus nivolumab combination and was given for 2 cycles. And then if you had uh, a great response to treatment, that was it. If you didn't have a great response to treatment, then you received additional treatment with either immunotherapy alone or targeted therapy. And then in the second arm of this treatment, you had surgery first, followed by immunotherapy. So kind of complicated, in the way this trial was designed. And one of the things that was that we're all facing a challenge with is trying to figure out what exactly happens when you have the immunotherapy first and then have surgery. Like when that lymph node is taken out, you can see a lot of dead tissue, but you can also see something called fibrosis, meaning like a lot of fiber in your lymph nodes. You could see melanophages, or where a lot of immune cells are eaten up and or eat up cancer cells and acquire the pigment. And you can have necrosis, which is just dead tumor tissue, or you can have on the extreme right viable tumor, meaning that the tumor looks like it's still viable. And so, what the Nadina study showed about a year and a half ago was that the doing the upfront immunotherapy was a lot better than doing the upfront surgery. And in terms of your possibility of recurrence, doing the immunotherapy upfront, that's that blue line on top, was much better than doing the surgery first, followed by immunotherapy, and the hazard ratio, which is just a way of telling how much better you're doing, was originally reported to be 0.32, meaning it was 70% better to do the immunotherapy upfront. And in Berlin, just a few weeks ago, the investigators updated that and they said the hazard ratio was 0.4, which is just another way of saying it's 60% better. So still better, but maybe not quite as the magnitude of difference was a little bit less than what was reported initially, which sometimes can happen as trials get longer follow-up. And then if you look at the safety of the two arms, like which was better and which had more side effects, clearly doing the neoadjuvant or doing the immunotherapy first arm had more side effects. And it has to do, I think, with the type of immunotherapy that you're getting. And so if you look at grade 3, just focus on this one line, uh, that was about 50% in the neoadjuvant arm versus 40% in the adjuvant arm. And then if you look at the serious grade side effects that are related to treatment, it's about 2 times more. It's 31% versus 15.9%. So you're paying in some sense for the higher response in terms of having more side effects. And that just shows the surgery related side effects and then the treatment related side effects. So what you're seeing here is that the surgery side effects are actually lesser in the neoadjuvant arm, a little bit lesser, just because the surgery is easier once the tumor is shrunken down, and so you're having a little bit less side effects. And then continuing with the study, what they also showed is, like, if you look at that top green line, if you had a great response in terms of pathology, like complete destruction of the tumor or only very little tumor left, pretty much uh, those patients up to 2 years did pretty good in terms of their having a really low recurrence risk and a really low distant metastasis free survival. And they also showed some interesting biomarkers. And I'll skip this because I think Dr. Lee is going to talk about this a little bit more. But uh, but basically, there were ways to tell who was going to respond well and who was going to respond okay. And so another trial that was discussed at ESMO was this trial that showing 3-year survival with a similar approach using pembrolizumab. And so in this trial, there were two arms in the trial. One arm, you got 3 cycles of pembrolizumab or Keytruda, followed by surgery, followed by 15 cycles of pembro, or you got surgery first, followed by 18 cycles of pembro. So the same amount of immunotherapy, but in one case you got 3 cycles right before the surgery. And again, very surprisingly, the doing the pembro before surgery seemed to give you a lot of benefit, and you could see that the hazard ratio was 0.67, which is a pretty amazing number given the fact that you're doing the exact same treatment in both arms. It's just who is getting it's just changing the order of treatment. And then if you look at recurrence risk, again, the doing the immunotherapy first was much better. And you could see that the hazard ratio is 0.42, which basically means that you got 60% benefit from doing just 3 cycles prior to surgery. And then what was also kind of surprising, to me anyway, was the fact that overall survival, was improved by doing the immunotherapy first prior to surgery. So you look at that blue curve there, it looks like it's the hazard ratio was 0.66. And this was very interesting because in many cases, these types of trials have not been able to show that you live longer with treatment. They've shown that the recurrence is better, but here we're beginning to see some sign that this is a big benefit. And I'm going to skip over this slide just because I think Dr. Lee is going to talk about this some more. But here's where things look like in terms of doing the neoadjuvant treatment or treatment prior to surgery. We talked about the NADINA trial that was done in Australia and Europe. SWOG 1801 is a trial that was done in the United States, uh, just using Keytruda or pembrolizumab by itself. There was another study done with this combination of Opdualag in the prior to surgery, or also known as Rela-Nivo prior to surgery. There was a study done at Mayo Clinic called NeoActivate, where they used a different immunotherapy agent called atezolizumab plus TIGIT, and then there's EA 1694, which is another trial using Keytruda plus this TLR drug. And one of the things that you can see is that pretty much all of the trials, you're getting somewhere between 50 to 60% response in terms of your pathology. So that is pretty encouraging, and that tells us that uh, a lot of people are probably going to do fine, even if you don't do much additional beyond the neoadjuvant treatment. And then again, I'll try to skip through these slides quickly. Not on the not so good, on the not as positive note, some of the trials that have been reported now showing really long-term follow-up are not showing the same benefit in terms of overall survival. So one of the trials was this trial looking at nivolumab or Opdivo for patients with stage 3 or 4 melanoma. And this paper was just published in the New England Journal of Medicine. So showing a nice benefit of nivolumab over ipilimumab or Yervoy. So this showing a nice benefit and a hazard ratio 0.76, which basically translates to about a 24% improvement. And so this looks good. But then if you look at the survival, which is the bottom line for studies in oncology, if you look at the survival, you could see that these curves are very, very close to each other. And Yervoy is an older type of immunotherapy. Nivolumab is a newer type of immunotherapy. So there were high hopes that the nivolumab would be superior, but that's not what we see in terms of survival anyway. And if you look at the side effects and subsequent therapy, you could see that, not surprisingly, a lot of the people who progressed on the nivolumab arm got different types of treatments as compared to the folks who did the same thing on the ipilimumab arm. Uh, and Another study, also not as positive, was looking at the Opdualag combination. It's called Relativity 098. And here, patients were randomized to either getting just the Opdivo or Opdualag. And you don't have to be a statistician to see that there is not much difference between these two curves. So with that, I'm going to conclude and let Dr. Lee talk about this. Yeah, but definitely we're not seeing a lot of survival differences in terms of preventative immunotherapy. But one thing we are clearly seeing is that doing the immunotherapy up front prior to surgery seems to be clearly better than doing it later. So let me end and let Dr. Lee take over.
Thank you, Adil, for the introduction. So I am a melanoma oncologist, physician scientist at UCSF. In addition to seeing patients, I run a lab focused on how to make immunotherapy better. So I will continue updating you on some of the findings that Dr. Daud has shared, but also kind of give you some insights on what we're doing at UCSF scientifically. Adil, can you see everything okay here?
Yeah, I can see nice and clear.
So to take some of the main take-home points from Dr. Daud's talk, a while back, melanoma oncologists really, when people have a melanoma, the standard of care was to really get the tumor out. And when immunotherapy was approved, people realized that these drugs can stimulate the immune system. So we would take the tumor out and assume that there's some residual tumors that are still left in the body, in the person, and we will give you at least a year of immunotherapy sort of to help your immune system clean things up. However, when doctors and scientists started looking into how this drug actually works, it's essentially a living drug. What we realized is two main things. The first thing is the immune system actually needs to see sort of its enemies to be able to educate and train its fellow sort of colleagues to be able to recognize the foreign looking protein. So that generated the hypothesis that maybe some more cancers in the body can actually help educate the immune system. And the second thing that I realized is that it turns out the more funky looking proteins that the T cells recognize, the more diverse that they are able to recognize the tumor's presence. And that can potentially help cure and sustain the cure a little bit better. So this is kind of a schematic that is on a really good journal here, saying that essentially when you do adjuvant, which is immunotherapy after surgery, the tumor has been removed, and when the drug comes in, there's not that many immune cells contacting and seeing T cells and being sort of educated and trained and having formed memory by them. But if you do give let's say the Keytruda or the Ipi-Nivo, Yervoy and Opdivo first, you actually bring in a lot of the T cells, make that area almost more inflamed. And that is why some patients initially will actually see their tumor get startled that their tumor initially gets bigger, but that is actually sort of the immune cells ballooning the tumor up. And actually, as the immune cells do its job, it's naturally evolved to actually be able to educate its compadres to actually recognize that tumor more. So that at the end results in a more sustained memory at the end of immune cells. So it's a whole new world. I asked ChatGPT why immunology, why neoadjuvant immunotherapy works better. And surprisingly, it actually did all right. So this is what it gave me. Let me click on this button. So why neoadjuvant checkpoint immunotherapy work better than adjuvant? So it gave this analogy. I did put, to be fair, I did put explain it to a 8-year-old or a 10-year-old or something. So it gave you this picture of one tumor adjuvant immunotherapy, just a couple of clone soldiers, and then if you give immunotherapy first, you really train a whole bunch of clone soldiers with different recognition, and then resulting in a more diverse potential for cure. So how does that translate to what's relevant to you, our patients, right? So what this means that for not just melanoma, for many cancers where immunotherapy works, I think the standard of care, because of the biology, I will show you the data that seems to suggest it's a very rigorous, solid data that a lot of immunotherapy responsive cancers would move towards this standard of care, which is we will give you immune therapy while the tumor is still there before surgery. Now, this is for even stage 3, high risk stage 2s, where you just have tumor at one area of the body. And then we will not wait very long. We will wait just enough, maybe 2 cycles of immune therapy to get the T cells in there. And when that happens, we plan that surgery to take the tumor and that sentinel lymph node where the immune system is being educated by the tumor antigen, will take the lymph node and the tumor out. And after that surgery, the lymph node goes to the pathologist where they can actually examine if the desired effects has been achieved. So hopefully most patients will get this result where you see the darker blue, almost black dots are like melanoma. Essentially, that would be when someone has their cancer in the lymph nodes, and then if it's actually cleared, you can see how pathologists can see that this now lymph node looks like it's just lymph node tissue and immune cells, and there's no longer the melanoma cells. So that's kind of what we would try to do. And this is a schematic, again, of how a pathologist would score your tumor. So a major pathological response. So this is kind of the main take-home point number 2. The remarkable data is that we actually didn't expect the correlation would be this strong. But if you have a major pathological response, that means when you got the neoadjuvant immunotherapy, surgeons cut it out, and the lymph node goes to be examined. If you have 0 residual viable tumor cells, or 1 to 10 only, and the rest is all immune cells, your survival, your ability to have a sustained, potentially curative response, this green line here, and there's now been about north of 3 studies that all show if you have that major, very brisk pathological response, your potential of having a complete response is over 90%, and your survival out to years is also very high, over 90%. Now, the part that the field is actively working on that needs to grow is, now, obviously, not everybody is always going to be fortunate enough to be in this category. What happens when you are a partial responder or non-response? And some scientists will call this kind of more of a cold tumor phenotype. And as you can see here, these patients did not get as brisk of an immune activity in that initial immune education phase. And their survival curve, this is percent of patients, and time on the x-axis is a little bit lower. And when you really don't have any response, then the risks are really, really higher. So this is still a very challenging case for oncologists. But what we're starting to do is we're really understanding a lot more on how this works. And just to recap some of the science that brought us here is that all of the neoadjuvant studies, right, they looked at why T cells were able to do a better job when the cancer was still there. And then why this is better than giving it after. So it turns out 2 things were learned. One is, like we were saying before, if you give an immunotherapy when the cancer is there, it brings the T cells to the area. And what happens is T cells work by detecting a specific foreign-looking antigen and then replicating itself. So when you see on the y-axis this word clonality, this really basically means how many different kinds of enemies that the T cells are recognizing. So, and those that recognize a different enemy replicated itself to make more soldiers that recognize that enemy. So this is saying that the responders compared to non-responders before treatment versus on treatment, right? So responders clearly can recognize more enemies. Their TCRs have more diverse clonality. And this study is comparing, hopefully my cursor is showing up, or I'm just pointing to myself, but the middle one is neoadjuvant generates more diverse number of clones than adjuvant treatment. And then once again, the responders, which is no relapse, has more clones than the people that do have relapse. So it's been now repeated over and over again, this clonality story. And this is finally another story that it's actually meant to say 2025, Georgina Long, so not 2015. But 2025, they actually did this in a highly incurable form of brain tumor called glioblastoma multiforme. And that actually is traditionally non-responsive to checkpoint inhibitors. But when they did this to this kind of cold tumor, this actually started working like a melanoma. They gave 3 checkpoint blockades because it's a very cold tumor, brought T cells into the brain tumor and then resected it, and resulting in educated T cells and less tumor cells. And then that created a scenario where this patient with GBM was now living up to almost 3 years. So pretty impressive biology. So just to wrap it up in like a couple of minutes, in our lab, we wanted to see if this is true for stage 2 and 3 kind of cancers, what would happen if we deploy this type of strategy in patients, and this is sort of future looking, right? In patients where they have what we call oligometastatic disease, where there might be more than one tumor site, maybe at 2 tumor sites, one in the liver, one in the skin, but both sites are highly accessible by our surgeons. Would it be possible that we can do this kind of immune therapy first, even though you're at a metastatic stage, educate the immune system, but then still have surgeons cut the tumor out after immunotherapy? So this is sort of dogma changing in oncology. Typically, surgeons do not really operate on stage 4 cases because of the risk-benefit analysis, where stage 4 surgeries are usually not curative. But now we're using it as a tool to sort of improve the immunotherapy response. So in our preliminary studies, where we have a mouse liver metastasis model, what this is showing is when we are measuring the size of the skin tumor, we can actually see when the animal gets checkpoint inhibitor first and then have their liver tumor resected, we actually improve the mice systemic immunity, where this blue group here actually has their skin tumor doing better and actually started having the checkpoint inhibitor working better and actually curing the mice. This is like mini mouse PET CTs here. And in mice that really didn't get this kind of neoadjuvant approach, the liver metastasis actually had a lot of ability to sort of suppress the efficacy of the immune system. So in these various controls with different kinds of drugs, with PD-1 alone, where surgery afterwards, it actually didn't work as well. So we started seeing that it actually, the science might be catching up to be able to understand and sort of better deliver this. And the final point that I wanted just to mention in terms of developing future better treatments is that we need to address the unpredictability of immune therapy, right? So our patients know that their side effects, this is a good publication that highlights basically you can have any organ being attacked by your own immune system, and it's completely unpredictable. And in our lab, we actually now can implant experimental tumors in different areas of the animals and test the checkpoint inhibitors efficacies, and we can see that there's clearly big differences in how these different organs impact the mouse's immunity. So liver seems to make things work less, bone mets work less, kidney, lung, and skin, subcutaneous SQ skin actually make checkpoint inhibitors work pretty well. So different tissue sites clearly have different response. And we don't think traditional labs that we do right now that are standard of care, like LDH, inflammatory markers, and measuring your protein or your blood cell counts, can actually be sensitive enough to detect these changes that may help doctors, change their plan or plan your specific personalized immune phenotype faster. The next phase, probably the next 5 to 10 years of this field, really will be seeing if we can get real-time immune cell data, which some of you know, gone through our consent process that some of our patients are already providing us, research samples that we can study their immune system. And some of the interesting in the last couple of slides is just to see that we can already have a tool that's sensitive enough where the red dots are patients with liver mets, and their immune cells look significantly different than people without liver mets. And as well, the responders look different than non-responders, and we are getting a statistically important and significant threshold to actually know where to draw that line and helping us predict the results. And finally, what's really cool is when you look at this ability to be able to predict results to the next best available technology, which is circulating tumor DNA on the lower right, and the old school technology, which is imaging here, you can see that on the upper right, which is our immune profiling capability, it actually tracks very well with the circulating tumor DNA's patterns. And in fact, if you look at some of these dates here, the immune system may actually be a sort of a canary in the minefield effect to be able to predict even earlier than ctDNA, because your immune system, who's surveilling your body, actually can, they're responding and changing in real time to foreign proteins. So that might be the most sensitive way to detect it. So kind of thinking about generating an immune telemetry as someone gets neoadjuvant treatment. And if we know that their immune system needs even more boost after surgery, we would give that person escalated therapy. But if someone clearly has a lot of effects, then we would de-escalate and the therapy to decrease the side effects and be able to have this thing titratable. So not everybody gets one size fit all 1 to 2 years of immune therapy. So that's basically sort of my update there. And I'm going to pass this on, the baton to Dr. Tsai, who's going to talk about the next phase. Thank you.
Thanks a lot, James. I'll let Dr. Tsai and Dr. Amouzegar talk about later stage melanoma and all the exciting developments going on there.
Thanks so much, Adil and James, for those really exciting updates. It's always good to see what's going on behind the curtain in terms of our clinical operations. So I would love to take this opportunity actually to introduce Dr. Afsaneh Amouzegar to kind of take that forward and talk about drugs that we use more often for metastatic melanoma. So we're out of the neoadjuvant adjuvant field now, talking about treatment for more widespread disease. Dr. Amouzegar, take it away.
Perfect. Thank you so much for the kind introduction, Dr. Tsai. Let me just take a quick moment to share my slides. And I hope it's looking okay on your end.
Looks perfect.
Okay. All right. Hi, everyone. Thank you for being here today. My goal is really to share an overview of how we treat metastatic melanoma today, and building on Dr. Lee and Dr. Daud's presentations, we want to talk about how far we've come in improving outcomes for patients with metastatic melanoma. So first, let's start by talking about what metastatic melanoma actually is and why it's such an important issue to discuss. So when we say metastatic melanoma, we mean melanoma that has spread beyond where it originated on the skin to other parts of the body that can include organs like the liver, lungs, or brain. Metastatic melanoma actually used to be very difficult to treat. Before 2011, most of the available therapies had very limited benefit, and there were significant side effects associated with these therapies. But good news is that now the story has completely changed. In the past decade or so, we've seen major advances that have really transformed melanoma into a disease that for a lot of patients can be managed long term. So this slide here shows how the landscape of melanoma really has changed. So you see that before 1998, there were very few effective options. Between 1998 and 2011, for over a decade, there were no new drugs that were approved. Then came the turning point starting in 2011, and that's when we started to see a wave of new treatments. Immunotherapies like ipilimumab and pembrolizumab, targeted therapies for patients who have specific mutations in their tumors. And now we have more than a dozen approved drugs for metastatic melanoma. So what do these treatments actually look like in practice? So today, most of our patients with metastatic melanoma are treated with one of these two main approaches. The first is immunotherapy, which helps the body's own immune system recognize and attack melanoma cells. These are medications like pembrolizumab, nivolumab, and ipilimumab. The second bucket of our treatments are targeted therapy. And the way these work is that they work in patients whose tumors have a specific mutation called BRAF. These drugs basically block the pathway that leads to uncontrolled growth of melanoma cells and stop the cancer from growing. For most of our patients, actually, immunotherapy is the first line approach, but the right treatment really can vary from patient to patient depending on different factors that I will talk about in a little bit. So looking at how immunotherapy works, our immune system is constantly surveying our bodies for things that just don't belong, like infections or abnormal cells. Cancer cells, on the other hand, are very clever. They can hide from the immune system by using these quote unquote checkpoints, which are like breaks that the immune system, that basically stops the immune system from recognizing the cancer cells and attacking them. The immunotherapy medications, like the anti-PD-1 or anti-CTLA-4 antibodies, they release these breaks, basically allowing the immune system to recognize and destroy cancer cells again. Every patient situation is unique. So when our team designs a treatment plan for you as a patient, we consider many factors. It's not just the tumor itself, but the person as a whole. Things like whether someone has an autoimmune condition, if they have any issues with their heart, kidney, or liver function, if they have brain metastasis or not, their LDH levels and overall fitness, they all play a role. We also take into consideration how fast the disease is growing and importantly, patients' own preferences. So this slide here shows one of the landmark studies that changed how we treat metastatic melanoma. This was the Checkmate 067 study. What this study did was that it looked at the combination of two immunotherapies, ipilimumab and nivolumab, which from now on I'm going to call Epi-Nivo, in comparison to Epi or Nivo by themselves. And really what they wanted to see was that how long did patients live after starting treatment? So what you see here are the survival curves, and each line really represents how patients did over time. So the higher the line, the more people were still alive at that point. So the most important takeaway from this trial was that now we have follow-up data for 10 years now. So 10 years after the study started, now we have patients who responded to combination immunotherapy. About half of the patients did respond. And those patients have long-lasting and durable responses, some of them even doing well 10 years after their initial diagnosis. And this is very significant for a disease that had a very poor prognosis about a decade ago. So for a long time, brain metastases were one of the toughest challenges in melanoma, but now we know that combination immunotherapy such as epi and nivo can be effective even in patients with brain involvement. So the Checkmate 204 was the study that looked at the combination of epi nivo in patients with brain metastasis. Here on, you see two sets of curves. The one on the left is showing patients who did not have any symptoms from their brain metastasis, and the curve on the right shows patients who were symptomatic from their disease. So what we saw was that in patients without symptoms, meaning patients who were only diagnosed with brain metastasis based on their scans and were not experiencing any neurological symptoms, more than half of them experienced a shrinkage in their tumor or even tumors disappearing. And many of those responses have lasted for years. In patients who did have symptoms at the time of their diagnosis, that benefit was more modest, but still very meaningful and something we could not have achieved before the era of immunotherapy. One of the new immune checkpoint inhibitor combinations that was approved in 2022 was Opdualag, which combined nivolumab, which we talked about, with another agent called relatlimab that targets LAG-3. So LAG-3, if you remember the PD-1 and CTLA-4 being these quote-unquote checkpoints, LAG-3 is another recently discovered checkpoint on immune cells. So this combination was studied in patients with metastatic melanoma, and it showed that patients who got this combination were disease-free and alive longer than the ones receiving nivolumab by itself. It also had a very favorable safety profile with fewer side effects compared to combination epi-nivo. So one of the questions in the field has been, okay, if we have a patient who has a BRAF mutation, how do you sequence immunotherapy and targeted therapy, since both of them are options? Do you start with targeted therapy or you start with immunotherapy? And this was what the DreamSeq trial tried to answer. So they compared two treatment orders in patients who had advanced melanoma and whose tumors had a BRAF mutation. So one group started with immunotherapy as epi-nivo, and then they switched to targeted therapy if needed. And the second group started with targeted therapy first, then switching to immunotherapy. And what we saw was that people who started with immunotherapy lived longer. So about 20% more were alive at 2 years compared with those who started with targeted therapy. And with longer follow-up, that survival advantage has continued, which really shows that there's lasting benefit for patients who begin with immunotherapy. So for most patients with a BRAF mutation and no urgent need for quick tumor shrinkage, we usually recommend starting with immunotherapy, since that's the treatment that offers the best chance for long-term survival. But the situation could be very different if somebody's cancer is growing very fast, or let's say it's causing severe symptoms, then targeted therapy can be started first to just get rapid tumor control and transition the patient later to an immunotherapy-based approach. There is a lot of excitement in melanoma research right now. Investigators are exploring new combinations of checkpoint inhibitors, new immune targets like TIM3, TIGIT, and really ways to make treatment safer by predicting who might experience side effects. We're also seeing the rise of cell-based therapies where a patient's own immune cells are modified or expanded to attack melanoma, which Dr. Tsai will speak about, as well as mRNA vaccines designed to train the immune system to recognize cancer. So one of the key challenges with immunotherapy is, as we mentioned earlier, is managing the side effects it can cause. I wanted to briefly mention a recent study that was presented at our annual oncology meeting earlier this year, which asked the question of whether adding an IL-6 or interleukin-6 blocker to combination immunotherapy could reduce side effects while maintaining an anti-tumor response. So interleukin-6 or IL-6 is a type of protein that sends a message to the immune system, and it can be implicated in inflammatory responses. And anti-IL-6 medications block that molecule and prevent it from sending that inflammatory signal. So in this study, they looked at a combination of Opdualag plus ipilimumab, so basically 3 types of immunotherapy combined together, pretty intense regimen, and combined it with an anti-IL-6 medication called sarilumab. And about 64% of patients had their tumor shrink or disappear. And the side effects at 6 months of follow-up happen in only 12% of patients, which is a much lower rate than what we expect with this type of strong immunotherapy combination. Having said that, it is still very early. This is a stage 2 trial and more research is still ongoing, but it does represent hope for future and safer treatment combinations. And lastly, recent studies have also focused on other subtypes of melanoma, for instance, mucosal and acral melanoma. These are the subtypes that usually start in places like the palms or the soles or from mucosal surfaces inside the body. And these subtypes behave differently from typical skin melanoma. They often do not respond as well to standard immunotherapy or targeted therapy. And one example that I've included here is a recent phase 1 study that looked at a new compound called IBI363, which combines checkpoint blocking with immune activation in a single molecule. And the early results that were presented in our annual oncology meeting earlier this year were very encouraging, showing that this was a safe compound and well-tolerated treatment. And now we have an active and enrolling phase 2 trial that is specifically looking at the efficacy of this compound in patients with acral and mucosal melanoma. So take-home points are that immunotherapy remains the backbone of treatment. Targeted therapy works quickly for patients with a BRAF mutation, but long-term control is often better with immunotherapy first. New combinations continue to emerge for patients with metastatic melanoma. Personalized decisions do matter. Your treatment plan really depends on a lot of factors, your baseline health, how fast your cancer is growing, and your mutation status. And clinical trials are the main key in advancing the field. And with that, I want to thank you for your attention, and we'll pass it on to my colleague, Dr. Katy Tsai, who will talk more about our clinical trial portfolio.
Hello there, I'm back. Thank you so much for the reintroduction and for the warm handoff for me to talk more about clinical trials. Let me go ahead and share my screen. Please let me know if it's the correct screen that's showing up.
Yes.
Perfect. So I am going to dive right into things. You've already heard a lot about clinical trials from the previous speakers, and I feel like even before I became an oncologist and involved in clinical research, it always seemed like a very kind of abstract thing, this black box of clinical trials. And, I can only imagine what it might feel like as a patient who might be considering clinical trials or maybe not even considering clinical trials yet, but wanting to know more about that. So the next 10 minutes or so are designed for you. So my goals really in the next 10 minutes are twofold. So number one, to provide an understanding of what clinical trials are. And the second goal is really to help provide a framework of how to think about clinical trials and whether one may be an appropriate fit for you or not. As Dr. Amouzegar just mentioned, really kind of every treatment we use today, all of these lines, these charts that we've been showing you show treatments that we use because they have at some point been through a clinical trial. So something really important to acknowledge in our field. So let's just really start off by asking the basic question of what is a clinical trial? I think that at the heart of it, I would divide trials into two main categories. There are more, but just to start off with the basics, on the one hand, we have what we call observational trials or studies. So these are research studies that only collect information over a certain period of time. There's no particular intervention or treatment that is involved in these observational trials. An interventional trial, on the other hand, is a research study that tests new ways to prevent, detect, or to treat a disease. And depending on the trial, of course, that treatment can take several forms. It can be a medical treatment like a drug. It can be a surgery. It can even be a behavioral intervention. And these studies really are designed to test these new approaches to see if there is a better way of doing things in what we currently consider as standard of care. So let's dive a little bit more into interventional trials, since I think for most of you, this is typically what we think of when we think about drug trials for melanoma. So let's drill down a little bit more into interventional trials. So what is an interventional clinical trial? And I think really at the heart of it, as I was trying to put together these slides for you today, at the heart of it really is, what does it take to turn an idea to fight melanoma and to bring it to a patient? So not a straightforward path necessarily, but I tried to break it down here into the following categories. You can see them in blue up top here, consisting of preclinical research, then clinical research, final data analysis, and then thinking about regulatory approval and follow-up. So really, I think the point that I wanted to make here is that the idea, before it even gets to being tested in people and taken through the approval process, that there's a lot of work that's done beforehand. There are protocols that need to be written. There are tests that can be done in a petri dish or in an animal as proof of concept, really, to show that something, that this idea, this thing, could potentially work to fight melanoma. If all of that seems promising, only then does it get taken into people, or what we call clinical research. So here, of course, there are various steps designed to look at safety and appropriate dosing of the drug in question, trying to figure out the side effects and how effective that particular drug is. And eventually, of course, the hope is that if this remains promising, to be able to compare it to existing drugs to see how it stacks up in terms of efficacy and toxicity against standard of care. After all said and done, if that final data analysis appears promising, then the hope is that a certain drug would receive regulatory approval and then get taken into wider distribution so that more people can access this particular drug. Even after approval, there is a lot of follow-up that happens behind the scenes to see how the drug is performing in a much larger pool of patients. So hopefully that lays the groundwork for the basics of what an interventional clinical trial is trying to do. And I'm going to drill one step further down into this clinical research portion here, because you have heard probably in your own reading from friends, family, and from us earlier in this webinar about the different phases of a clinical trial. So I did want to spend just a couple of minutes explicitly discussing that here. As you can see, in general, the life of a drug or the phases of a clinical trial are split into 4 phases with slightly different questions with slightly different questions being posed at each step. As you can see for a phase 1 study, typically here these can be first in human studies, and so therefore hopefully it goes without saying that the majority of the focus is on drug safety and appropriate dosing. And as you can see on the bottom half of these rectangles, phase 1 trials do tend to be the smallest pool of patients, and then slowly increasing in size as we move on in phases. A phase 2 clinical trial, now that safety has been established, tends to focus more on how effective that particular drug is in a certain pool of patients. So here the pool of patients is slightly larger in number in general, up to 100 patients or sometimes even smaller than that. And then once we have this body of data building on drug safety and drug efficacy, then we have a phase 3 trial, which generally will compare the new treatment to an existing standard of care in a pool consisting of hundreds of people. Phase 4, as I mentioned earlier, generally is when a treatment is approved and available, and there is longer term follow-up in a much larger pool of patients being investigated in terms of longer term efficacy and longer term toxicity. So again, each phase is trying to answer a slightly different question, and patient safety is paramount and is being checked at each step. So why even consider a clinical trial? So here I'm going to repeat a slide that Dr. Amouzegar showed us earlier. And just to drive home the point really that the breakthroughs that save lives today exist because people before you volunteered to participate in clinical trials of these drugs. Within melanoma, you can see that the field has expanded enormously quickly over the past 1 to 2 decades. Certainly, I think during my career, it's been amazing to see the variety of different drugs that have been approved. And even more importantly, as you've seen with the previous talks, that the same drugs or similar drugs of the same category have pretty distinct roles depending on which context of treatment they're being used in. That is, how is the drug performing for treatment of patients with widely metastatic disease versus how is it performing when we're using it in the neoadjuvant or adjuvant treatment settings. So all very exciting. And now that we're in 2025, looking forward to seeing more, hopefully more approvals coming our way. In terms of how to find a trial, so I think that this is probably a pretty select population. Here at UCSF, if you found us, hopefully you share our enthusiasm and goals of driving the field forward. So obviously, your oncologist, your cancer treatment team, is an excellent place to start. Here at UCSF, we're lucky, to be plugged in nationally and internationally to melanoma colleagues who are doing extraordinary work. So if you have questions, please ask us. In terms of UCSF specific trials, you can see here that I've put in a website for UCSF specific trial finding purposes. National databases, of course, despite recent funding issues, also hopefully remain an excellent go-to. I note that because clinical trials are so dynamic, they open, they close, maybe they are open, but they're no longer enrolling new patients. So whatever you find about the status of a trial online, please take with a grain of salt. The listings can sometimes be up to date, but at least it gives you an idea of what's out there. And when in doubt, bring in trial listings, right? As clinical researchers, as medical oncologists, we can help you to interpret what seems relevant versus what may not be as appropriate for your particular clinical situation. And of course, if you need help outside of your immediate resources, friends, family, oncology team, there are foundations, AIM at Melanoma, Melanoma Research Alliance, that have online trial navigators as well that are available for your perusal. So let's say you found a trial, or at least you've gotten to the point where you're thinking about joining a trial. What can you expect? So first and foremost, I think there is a somewhat intuitive expectation that once you sign consent, it means, hey, presto, you can start the trial. And I always like to emphasize that signing consent actually doesn't mean that someone is automatically part of the trial. What it actually means is that it kicks off what we call an eligibility period or a screening period. The length of that period can depend on the trial. In general, it can be anywhere from 2 weeks up to 4 weeks. And that is a time designed for us as your study team to get all of the testing and any procedures necessary to ensure that it's safe and appropriate for you to be on that clinical trial. Unfortunately, there's not one standard set of eligibility across all clinical trials. Depending on the clinical trial, what phase it's in, the study goals, eligibility can vary according to the trial. So really important things to bring up with your study team. Also important to emphasize is what I like to call the hassle factor, or some degree of inconvenience. Now, keep in mind that sometimes the drugs that are available in clinical trials are novel, and therefore safety really is the most important thing that we need to track for our patients. Therefore, there may be additional things that we ask you to do that you wouldn't necessarily have to do if you were getting something that was already FDA approved, already standard of care. Those hassle factors, as I like to call them, can range from anything to extra visits at UCSF, extra blood draws, or more frequent scans. So something to keep in mind. On the upside, what you do have is very close monitoring from the study team, from both all of the oncologists that are involved in the study, from the study team at large, and you are assigned as a clinical trial participant, a clinical research coordinator, who is a very important member of our team, who is your primary point of contact during this entire process, both before the study and when someone is already enrolled to a study. Also worth keeping in mind that your insurance, of course, needs to provide authorization for you to participate on the trial. That's something that's built in as our usual workflow. Helpful to keep in mind that there are routine things that will be billed to your insurance, but very often study-specific care and, of course, the study drugs, these are things that are often covered by the sponsor of the trial. So as we're drawing towards the end of this talk, I wanted to address a couple of kind of common questions or maybe misconceptions that we often hear about clinical trials. So number one, I just wanted to address the elephant in the room, or maybe the guinea pig in the room in this case. Will I be a guinea pig? And absolutely, the answer is no. There are very strict safety rules that protect our clinical trial participants. Any site that's participating in a clinical trial obviously is subject to audits, both by the sponsor of the company, by regulatory authorities. So definitely there are processes and workflows for everything that happens on a trial. Will I get a placebo? And here I would say it depends on the study design. Obviously, to keep clinical trials ethical and reasonable for patients to enroll to, there may be a placebo, but only if it is considered ethical. That is, it would be considered as something as a reasonable standard of care. So please don't take it as a given that you may get a placebo if you enroll onto a clinical trial. Another common question, are trials only for advanced melanoma? I think sometimes there's a perception that trials are there only as a safety net, only in situations where someone has exhausted all possible other treatment options. While certainly that situation may come to be, I would also say it's not universally true. Different trials may be testing drugs at different earlier stages of disease, for example, taking something from the metastatic setting into a neoadjuvant or adjuvant treatment context. This is also another common question we get, how soon can I start? I can start tomorrow, I've already signed consent. And hopefully I've already addressed that, the answer to this earlier in terms of talking about that eligibility period after you sign consent. Last but not least, one common question, will I have to stay on the trial until it ends? And here definitely the answer is no. These are voluntary participation. And someone can choose to stop their participation in a clinical trial at any time. So I know that this is a bit of a word-heavy slide, so I won't read it out loud, but I just wanted to put this up here in terms of questions that you may want to consider asking your study team as you're considering a potential clinical trial as part of your possible regimens. I will highlight here that kind of having some sense of what tests and procedures you would need to have done during your eligibility period. Often it can feel like a blur. Blood work, imaging tests, biopsies. And so I think sometimes having a clear idea of what you actually need to, what what your study team is actually asking you to have done beforehand can help reduce some of that kind of black box or fear of the unknown going into things. So this is my last slide. Take-home points here really are that clinical trials have driven every advance in melanoma care since we started. That being said, participation is voluntary and highly regulated for safety. And while obviously as a trialist, I'm biased, and clinical trials are, they play an incredibly important role in what we do. They may also not be for everyone. So ask those questions, empower yourself. And all of this, of course, on our end is only possible with our, mighty group of clinical research coordinators and investigators. You can see here some of the events we've had as fundraisers and partnering with melanoma-specific foundations. So thank you for your time. I will leave it at that.
Great. Thank you, Katie, for that nice. I'll try to remember the trial guinea pig. You're not just a guinea pig, but you're something more than that. And let Dr. Tao talk about cellular therapies, which is like a super exciting new development in our field.
Great. Thanks for that introduction. Yeah, good afternoon, everyone. I'm honored to be here and speak to you all today. Really, truly, as a Bay Area native, originally from Oakland and a Berkeley graduate, it's a privilege to be back and provide care for the community that really helped shape me. So hopefully you guys can see my slides here. I can advance. Great. So an outline, keeping it simple, I'll be introducing a complex topic of adoptive cell therapy and talking about two forms of that type of therapy. So you've heard about amazing advancements today from our team, and the reality is that we're not satisfied. We are constantly asking ourselves what else we can do. And adoptive cellular therapies are one of those areas we think we can offer for patients that need more. So what are adoptive cellular therapies or cell therapies? Well, this treatment kind of still strikes me as science fiction that's come true, really. It's quite complex, but it involves collecting immune cells, usually T cells or the immune system soldier cells, from the patient. We then modify them or expand them outside of the body in a lab, and then they're adopted back into the patient's blood system to fight the diseases like cancer. And so there's three most commonly discussed types of adoptive cell therapy. Highlighted here first is the oldest, tumor infiltrating lymphocytes, or TIL therapy, as we call it. And it starts with a surgery and harvesting T cells from the tumor itself. And then that tumor is kind of broken down so that we can collect just the T cells that have been targeting the tumor. And then we grow them up so that we can give them back to the patient. And so here's a representative image of the process from a lab in the NIH showing here you start with the pigmented melanoma cells along with some other cells in the background. And then over time, the melanoma cells are dying off as you're enriching for the T cells that you see here at the end. And so this work is definitely very exciting. And it's the newest FDA approval for metastatic melanoma. It's the most recent of 14 approvals we've seen in the last 13 years or so. And while I said it's the newest approval, it's also one of the oldest therapies for melanoma. So I'll highlight here that actually development for TIL therapy began over 36 plus years ago at the NIH surgery branch. And so I'll show some data that came out from the NIH team. And so while I'm showing some data that has a lot of detail, I'll just kind of be highlighting some of the important concepts. And so this report showed an objective response rate of 56%. And so objective response rate being a way to say shrinkage of tumors greater than a specific cutoff that we set. And survival for melanoma was over 22 months. It was 28 months if you hadn't had PD-1 therapy, the prior checkpoint inhibitor therapy. But about half of that if you had PD-1 therapy in the past. And so it's notable to say that patients in this study were being treated in 2013 to 2018, so almost over 10 years ago, which is a long time in the context of considering how rapidly melanoma treatment is developing. Another important point to highlight is that for this top line, what we call CR or complete responders, those patients that had disappearance of their tumor, they went on to live and have a wonderful survival. And so Iovance is a company based out of San Carlos. They've been the ones that successfully commercialized this process that was developed in the NIH. They have a product now called Lifalusil or Amtagvi. And it was the one that was granted FDA approval based on this clinical trial, C14401. And you can imagine that there's a lot that goes into developing this from a specific group and a specific lab in the Washington, D.C. area, and setting it up so that we can standardize it for widespread use. And that's included processes like needing to freeze the cells. So that's one of the hurdles that they had to overcome. And that's why in this trial, we have kind of multiple cohorts and only a few of which went into the FDA approval analysis. And then another hurdle has been that most of the patients treated when this trial was done received immunotherapy before. And so despite that, we see still an impressive objective response rate of 31%. And that's each vertical line here is one patient and how their tumor changed from this horizontal 0 bar. And if it goes down, that's showing the shrinkage in tumor. And so many of them shrank and disappeared in green here. Many of them shrank below our cutoff of minus 30% change from baseline. But then even beyond that, we still have a large group that had shrinkage, maybe not as significant, but still some shrinkage in control of their disease. Another trial was done looking at TIL therapy from a group in the Netherlands and Denmark. And they put it up against in a randomized study, so something a little bit more rigorous in terms of testing. And I'll kind of just use this slide to highlight that while I've talked about TIL generally, it's a little bit more complex in terms of the sequencing and how things actually get done for patients. So patients usually come to the hospital about a week before they get the cells, and they're given 5 days of chemotherapy. So this is cyclophosphamide and fludarabine, ultimately to kind of clear some space for the cells that they're going to be receiving. Then after a couple of days of rest, they get those cells infused. And then soon after that, they get something called IL-2, which is like a stimulant or a nutrient for the T cells to continue to grow and proliferate to be able to fight the cancer. So an involved process. And in that study, I'll point out that they saw an objective response rate that was slightly better at 49%. And putting those studies together in terms of the TIL at the NCI, the FDA-approved product, lifileucel, and the randomized study I showed you from Denmark and the Netherlands, while not appropriate to kind of compare them, it's worth noting that there's obvious differences in the study, like I pointed out in terms of the decade in which they were done. The patients having sometimes more aggressive biology, we see represented by an LDH kind of as an approximation. But also the types of therapy patients had received in the past. So again, this is one of the newest FDA-approved therapies, the lifileucel. It is the latest one, and thanks to our team led by Dr. Katy Tsai's efforts here to get this up and running and ready to give to patients at UCSF. In terms of next steps, this company that produced lifileucel is also looking at using it earlier for patients. So even before they've seen immunotherapy, we could give the TIL in conjunction with immunotherapy like pembrolizumab. And so this is data that was involved in a phase 2 study and has become the basis for one of their phase 3 studies here. So moving back to adoptive cell therapy, a broad category that includes multiple types of treatments. We talked about TIL, kind of the oldest, but also newest FDA-approved therapy for melanoma. There's also CAR T cell therapies. People hear about this a lot in the context of certain cancers, specifically hematologic cancers like lymphoma. So not as relevant for our conversations today. But then there's TCR T cell therapies. And this is important to introduce because it's kind of the basis for a lot of the new exciting work and new clinical trials that we have going on in the melanoma space. And so in TCR T cell therapies, so TCRT cell therapies here, we take the patient's blood, actually, and we use that to collect T cells and engineer them with specific T cell receptors that basically are able to identify a target on the cancer. And so there's some important distinctions between these types of therapies, but in this picture here, the T cell receptor is shown by this kind of light blue complex here. And it's basically able to recognize the red piece, which is a part of the tumor, but it's also connected to a kind of like a cup that's holding it up and presenting it. We call that the HLA molecule. And so the T cell receptor actually has to be not only specific to this piece from the tumor, but also this HLA molecule that's presenting it too. And this has been a very important part of developing more therapies for patients because while CAR Ts and some therapies are only able to recognize proteins on the surface of cells, these TCR T cell therapies, by virtue of identifying this kind of complex, are able to identify the multitude of proteins that are inside of a cell. And so it kind of opens up the possibility to target more proteins from the tumor itself. It also brings up one of the most kind of main limitations right now for this therapy, which is that many of these T cell receptors are restricted to certain types of HLH or HLA, which are specific to different patients. So the one that's most clinically developed is one called HLA A201, A0201. And that one right now is expressed in about 20 to 40% of Caucasian Americans. So it's an area that we're in urgent need of developing more that can be more accessible for more patients. And that's kind of what I've been showing here. So in terms of areas of improvement, I think one thing I didn't highlight yet is that the TIL cell therapy needs to be manufactured from each patient after they have a surgery. And that period of time can be sometimes what feels like an eternity with 4 to 6 weeks, although there's processes working on making that shorter. Sometimes the product isn't successfully made. There's still the issue that patients have to go through a surgery that can be difficult and require recovery time. Patients have to be pretty strong and capable of, and in some ways we talk about their activity level because of the need to go through this long process. And it's a resource-intensive process. Additionally, from the scientific standpoint, we're looking at ways that we can improve upon the effectiveness of this therapy and also reduce the toxicity that comes with it. So let me move here to the areas that we can improve. So there's been efforts to look at collecting cells for patients, not just from a surgery, but maybe from blood collection, as well as allogeneic off-the-shelf products, which is an area of interest of mine. That would be the topic of another talk. In terms of reducing manufacturing time, we're working on optimizing all of this as well. And then there's ways in which we're looking at optimizing the effectiveness of those T cells, reducing the side effects of the other medications we have to give, like the lymphodepletion and the IL-2. And then there's strategies that are very creative looking at combinations. And so one of the specific TCR T cell therapies that is most clinically developed and up and coming is targeting something called PRAME. It's basically a marker on the majority of melanoma cells. As you see here in cutaneous melanoma and uveal melanoma, we have over 90% of patients having this target on their tumor, which has made this open up the opportunity for this therapy for all melanoma patients. In terms of this product itself, it's one from a company called Amouzegar that I've been partnering with and our team is working with now. They have a product called IMA203, and in some of their early clinical trial testing, they were able to show that for melanoma patients, again, these vertical lines showing going downwards, showing shrinkage of tumor for most of patients treated with this medication. And then here we see from each patient starting at the 0 line, their tumors are shrinking and they aren't growing back, and the control of the disease is very stable, with some patients still having further shrinkage of their tumor out many, many months. And so this has led to a randomized phase 3 study that's now open at UCSF. This is very exciting for our patients as well. There's also further developments in terms of trying to armor these products and these TCR T cell therapies to be even more effective. So here's just an example where they're trying to give it a special nutrient called IL-15 rather than using the other nutrient I talked about, IL-2. And then another effort that we've been working with Amouzegar on is a phase 1 trial that uses that product that you're talking about now called Amouzegar, also IMA203. They've just given it a name very recently. But adding to that a cell therapy enhancer or a mRNA vaccine, basically able to re-stimulate this T cell so that whenever we give the vaccine, the idea is that these T cells will be kind of reinvigorated. And so this is a trial that's very nearly up and running at UCSF as well. And then very recently, some data from the same medication and treatment, Amouzegar, for uveal melanoma patients, which has been historically one of the most hard to treat types of cancer. And again, we see very impressive reduction in all of the patients' tumors in this early trial. And so I'll end by saying that we are very optimistic and excited to continue pushing in this field of therapy for melanoma patients. And just like the first immunotherapies opened the door for several more developments that quickly followed, we think that TIL really just marks the beginning of a host of new therapies that will be approved in the near future. So thank you all for your attention.
Thanks, Derrick. Thanks for that quick tour through cell therapies. I know it's taken a lot of work, and yeah, we are excited to have you here. So next, I'd like to turn to Liz Gasper and Veronica Meraz, who are patients on clinical trials here at UCSF. And I know a lot of times people say like, hey, sure, all this stuff sounds good, but what is as Dr. Tsai was saying, like, what is actually involved in getting on these trials, and how much hassle and how much risk, and what exactly do you have to put up with? So maybe I'll ask Liz to talk to us about that first, and Veronica, if you could do too.
Hi.
Hi. See, you have your nice glasses on.
I know. Yeah. I'm only seeing one of you. So do you want me to just chat about what my experience
is? Just maybe if you want to take a dive into like what does it mean, and
Right.
just the hassle, the
Sure.
risk, what do you think when somebody asks you to sign a consent or talks to you about a novel drug?
Sure. Well, just real quickly, I had been undergoing immunotherapy, and I was one of those people that had a very bad reaction to it. So that was off the table to treat my melanoma. And Dr. Daud, with a lot of speed and effort, got me into this very exciting clinical trial that has worked very successfully for me in a very short period of time, and that my melanoma, my tumor's gone and stuff. I actually commute from Reno, but aside for driving to the Bay Area, which I like doing anyway, this has been very easy and very seamless. I get great care. The approval process was very quick. It was less than a month from the time that Dr. Daud mentioned the clinical trial to me to where I actually started taking the drugs. And they keep a very good eye on me. They check in video-wise as well as I go now once a month. But it's been a very easy and seamless, and most importantly, a very effective clinical trial. So I asked a lot of questions before I did it, because basically every single question Dr. Tsai had on her list, I asked. And anyway, I'm very happy I did it.
Hi, Ms. Gasper. I'm glad to hear about your experience. Nice to see you. I guess glad to hear that we've gotten to a seamless place. Can I just ask you, is there anything that you found surprising, or maybe in retrospect wished had been done that would have been helpful that maybe wasn't?
Yeah, I can't eat grapefruit, and I love grapefruit. That's the only food I can eat. I can't eat, they said. But no, and I'm I'm not trying to sugarcoat this at all, but no, it's been as expected, if and even better.
Yeah, thank you for sharing. And I think maybe just to acknowledge as well that depending on the trial, depending on the drug, as you mentioned, there can be prohibited medications or prohibited foods, in your case, grapefruits, because they interfere with how the study drug gets processed. So sometimes there are additional sacrifices, so to speak, that need to be made while you're on a trial. Thank you for sharing.
Yeah, but in my case, it was only grapefruit.
Liz, what about the hassle factor? I mean, that's not a simple thing to be driving that distance and, coming in on like back-to-back clinics and all that type of stuff. I mean, how is
Yeah,
that interfering with your life? I mean,
quite honestly, and I'm probably a little bit unique in that I have a consulting practice and I have clients in the Bay Area, so I'm able to work around that and friends. So it's it actually is a pleasant experience for me to drive to the Bay Area. As oh dear, I got to go to the Bay Area. One of the things I should mention, and it's a minor inconvenience, I have to track when I take my medication and keep records. But in my case, I take 3 pills twice a day, and I write them down in a log. So if if I didn't have any other reason to go to the Bay Area, sure, that would be a hassle because it's a 3.5 hour drive. But like I said, I use it as an excuse to visit family and friends and actually do business. So it's worked out pretty well for me.
and I see that Kendall is right. I see her shoulder.
Yes, this is
Hi.
my daughter, Kendall, who has been a great source of support.
So, Kendall, what's it like for you? I mean, I know you've had you know, some some difficult times, some
It's been a huge learning experience for me. I trying to understand how I could help or be of service, and working with the information that I was given from the great medical team. It's been amazing. I've been able to reach out to the medical team. Dr. Daud, it's very easy to communicate with you, and I appreciate you just teaching me a lot of things. And sorry. I'm thankful. Thank you.
I'd like to interject something, if I might, not to put words in your mouth, but Kendall, since she was a little girl, always asked why? And she has asked a ton of questions and they've all been answered, and they've all been answered to her satisfaction. And so that's taken a lot of your guys' time to do that. So that's another, I think, very positive thing of the clinical trial, in that any question that comes up, it's very easy to get a hold of you, especially through my chart and stuff, and seeing results of tests that we have through my chart. So anyway.
So what would be your candid advice to people who are considering clinical trials? What would you say to them?
Well, I think exactly what you guys had on the slide. Ask a lot of questions. We all have different lifestyles. We all have
Study
different,
and ask.
yeah, study and ask and make sure it'll fit in with your lifestyle, and challenge. I mean, we say, well, but wait, what about this and what about that? And the only way you're going to get to the right answer as far as whether the clinical trial is right for you is if you ask the questions of your medical care team and get those answers, and then you can evaluate. And then if it doesn't work out, you can always say no. And that was another thing I think that you guys made very safe for me, was that I never felt like, oh my God, I signed the paper and I committed to this, and that it was okay if I wanted to change my mind.
I think it's wonderful that it's a team. It's not just one person working on their own, it's multiple.
Yeah, yeah. So
that's huge. So lots of challenges, but sticking together, working together, studying and learning the new information and moving forward.
Yeah, I think that resonates, and it takes a village, as I like to say. But thanks for emphasizing that, because I think it's one thing for us to say, like, do you have any more questions? What other questions do you have? But I think quite another to hear you say it as someone who's actively on a trial.
Right.
Yeah. I see Dr. Amouzegar on as well, and also Ms. Meraz. I'm not sure if you're in a position to be able to unmute and turn on your video camera. But if you are, I just wanted to invite you to provide maybe some counterpoint on, or not, on what your experience has been like so far. Ms. Meraz, just in case you're having some technical difficulties, I'll just invite you to, if you look at the bottom of your Zoom screen, there are buttons to unmute yourself in case you're there. But if not, we can also pivot to other things.
So maybe we should open up to everybody for questions and ask. And so maybe I'll start off by asking Dr. Tsai, what is, sometimes people talk about phase 3 trials and phase 2 trials and phase 4 trials. What's been your experience in terms of the pros and cons from the patient's point of view doing phase 3 trials versus phase 2 trials?
Yeah, that's a great question. I mean, I think if you remember that kind of pathway I showed from phase, 1, 2, 3, 4, I think sometimes phase 2 has, I hesitate to use the word advantage, but these are drugs that have been shown to have some exciting data, from the preclinical phase already, and there's already some safety established with that. So I think sometimes in the phase 2 setting, it's a little bit more exciting, right? This is something that is relatively new and earlier on in drug development. It seems promising. It seems safe. Let's see how effective, let's widen the pool. We have some idea of how effective it is, a good idea of its safety. So let's see how it performs in a bigger group of patients. That's certainly not to knock, the phase 3 setting, right? Where I think maybe no less exciting, but just exciting in a different way. So in the phase 3 setting, it's a drug or an asset that now we have even more information about efficacy and safety. And when push comes to shove, how are we going to use it, right? How does it stack up against, what we might already have that is considered standard of care? So yeah, I mean, sometimes as us being in clinical research, I know that sometimes a phase 3 sometimes can seem, for lack of a better phrase, a little bit less sexy, a little bit less novel, but still definitely important. And if a drug has made it to phase 3 drug development, I mean, I think it's super exciting, right, in a different way that it might actually replace something that we're already all widely using.
Afsaneh, what about you? Sorry, what do you think is the most exciting development? And if you want to tell us about your move to San Francisco too, that's great too.
Yeah, no, I would say in terms of personal experiences, the biggest development for me has been moving to the Bay Area, which has been so far an amazing experience to also be part of such an inspiring group here and being able to really serve the community. Some of the advances that I'm really excited about is actually the cellular-based therapies. I think those are going to be very revolutionary, and I'm so happy to see so many new trials in development and a lot of them opening at UCSF. it just basically is a very interesting kind of a very exciting concept to be able to offer such a personalized treatment to patients, especially in a setting where some of our more conventional treatments may not work. So that's one area that I'm particularly interested in. I know that's one area that Derrick Tao is very focused on, and having him here on the team is also very exciting. But the other area that I'm personally interested in is maybe focusing more on that harder to treat population of patients that have brain metastasis or more advanced diseases involving the central nervous system, since that's always been a challenging patient population to treat. Although we've come a long way, there's still much room to grow and improve. And I'm hopeful that we would be able to start having more trials here at UCSF specifically targeted towards that patient population.
James, what about you? What's your personal like high from this year's scientific meetings, or you personally?
I think it's a big year for melanoma. It was sort of at a second phase of growth. I feel like after the initial checkpoint inhibitors that sort of broke the glass ceiling for immunotherapy for solid tumors. It was stagnant for a little bit, but now actual adoptive cell therapies are getting approved and coming online. mRNA vaccines are finally being associated with cancer instead of COVID. So that's all very good for immune therapy. I think it's a very hopeful, promising time. And also like the technological advancements with high throughput computing and AI. Our lab is already starting to use it. Big companies are really able to use it to start matching genomic and protein data with treatments. I think that's going to accelerate advancements and growth in cancer.
Katie, what about you? What's your personal high point for in terms of all the advances, and what are you going to be watching for in the next few months or?
Yeah, I mean, I think the theme or I think the overarching theme tends to be, it's an exciting new world for adoptive cell therapies for solid tumors. And especially, we saw from Dr. Tao some really exciting data for that in melanoma specifically. Yeah, I'm just also looking at the chat. Hi, Mr. Pelsinger, and I think you had a question specifically for Dr. Lee about TILs, but I mean, I think to Dr. Tao's point, while I'm happy that we're, getting on the bandwagon of being able to provide TIL therapy here, we're at the tail end of getting that operationalized here. It's kind of that first wave, right? I think TIL therapy in its current form has it's a big hammer for a big problem, as they say. It's kind of how yeah, it's operationally heavy, requires a lot of logistics and hospital stays and coordination. So I'm just really excited to see how we move on from here. There's already clinical trials, of arguably, well, hopefully, better, TIL therapies for our patients. So I'm looking forward to seeing more about that. Dr. Tao, any kind of piggyback comment on that, given the ematics trials that you've got a hand in and others as well?
Yeah, I think in a situation where, a patient isn't needing it now, isn't needing cell therapy now, the field might change quite a bit in just a couple short years, even, months in the sense of what trials we have available, and what those trials are bringing to the table in terms of advancing it. Ideally, not only from the standpoint of effectiveness, but also from the standpoint of lowering patient burden, lowering patient like side effects, making it more tolerable, also making it more efficient for patients too. So there's a lot of exciting trials happening in that space. To Adil's earlier question about kind of exciting therapies, along with what I'm passionate about, cell therapies, I'll mention also kind of mRNA vaccination. I think with what Dr. Lee said, the opportunities with AI, kind of higher power computing, it really allows academia to be in the game for and participate in a lot of not just discovery, but also kind of these therapeutic developments. So I know we're working with a lot of top scientists at UCSF to kind of develop this around this area of personalization, which I think academia does really well for our patients. And a setting like UCSF is very poised to be able to do that. So building out a personalized vaccination program for our patients will be very exciting in the near future.
We do have a couple of questions on the chat. I wanted to make sure if we could briefly go through them.
I see there's a question from Tom Bell, who's, I think, lived through some of these changes. I think he was on the Ipi-Nivo brain metastasis trial back in 2016. And it's been a long journey, right, Tom, since then? And yeah, to the point that Dr. Tsai was saying earlier, that participating in these trials, it makes sent our field or been such a huge advance for our field.
I'm so glad too that we've also gotten to the point where I think Dr. Amouzegar had a slide on this earlier. I mean, we're all used to talking about the drug interventions, right? Therapeutic trials to obviously treat someone's melanoma, shrink it down, get someone into remission. But also the flip side of the coin is interventions for immunotherapy toxicity, right? I think it's something we all struggle with in terms of how idiosyncratic or unpredictable these immune-related adverse events can be. Historically, a lot of what we do to treat these IRAEs, as we call the immune-related adverse events, has been just based on experience, institutional experience, personal experience. And I was really happy to see at the big oncology conference this summer that a trial for an IL-6 blocker got some airtime in terms of, granted it's a small phase 2, not a lot of patients, but really exciting to see that it can help hopefully reduce toxicity without compromising anti-tumor efficacy. So hopefully, more studies, larger studies will be following that.
Definitely. And as we get more approvals for immunotherapy for basically every cancer type, is even more reason and more important to start looking at ways to mitigate these IRAEs and toxicities.
Adil, I think you and Ronnie were just talking about some exciting new NRAS drug, right? That there's the question about any new advancements for NRAS. I mean, it remains to be a very challenging, generally speaking, less tumor mutational burden, so less responsive to immunotherapy at times. So we do use molecularly targeted therapy, and companies are getting very creative at designing more and more potent drugs that can actually act on the NRAS pathway. But yeah, it remains to be seen if the durability of these molecular drugs problem can be solved. My personal hypothesis is that I think this might be an opportunity where some of the cold tumor strategies where you potentially combine immunotherapy or use bigger combinations of immunotherapy up front in a neoadjuvant approach, and potentially combining with some kind of a vaccine, or bispecific engagers like Immatics type of a treatment might be needed. Because you would need to artificially bring in T cells to the environment. So obviously that opens TILs as well. But any other comments, guys, for NRAS specific questions?
I think there's the there's a new class of NRAS and KRAS and HRAS blocking, but mostly KRAS blocking drugs, like one that's made by this company called Revolution Medicines that's based in Redwood City. And that drug is being tested in pancreatic cancer and lung cancer currently. But it's showing a lot of promise in preclinical studies in NRAS mutant melanoma. So I think the one effort is to try to get a clinical trial organized with that Revolution RMC drug and other, they're called molecular glues. So those things are entering clinical trials and stuff like that.
I see, Mrs. Meraz, you've unmuted yourself. I'm glad to see you were able to join. Would love to hear from you if you're able to maybe briefly share your experience having been on one of our trials for the past month or two.
Well, so far so good. I've been feeling really good. I did start with the immunotherapy, and my first treatment went really well. And after the second one, I got really sick, and I'm not sure why, but was having difficulty eating, nothing sat right. And then I got approved for the trial, and so far it's been going good. I'm not as tired. I did have a couple of days where I was really, really tired and slept a lot. And other than that, I've been feeling really well. I'm up and doing things and feeling a whole lot better. So fingers crossed this is, going good. I was laughing at the slide because that's kind of the way I explain it to people. I said, my son is a big Star Wars nut, and I said it's like the force field. I said, it's got to go in there like the Death Star and blow everything up. So
No, glad to hear that you've been feeling well and doing well. One question that I wanted to ask you, how would you compare your experience being on a clinical trial compared to the previous treatments that you were getting in terms of the experience and the overall how you felt?
well, it was really scary at first, when they say it doesn't work and then you have to go to a trial. But the experience has been very positive. Things are explained to me. I don't feel like just a number, That was kind of my first thought, is that I'm just going to go in, they're going to, give me a drug and send me on my way. But step by step, people have explained things. The doctors have been very forthcoming in answering my questions. And that's the nice thing, is I do tend to talk a lot, but they're very patient with me, and they explain everything. And it's not just like, here you go, get on your way. So that part has been very, very positive. So
Thanks for sharing your experience. It cannot happen without our amazing clinical research team who walk you through all the steps that are involved in a clinical trial. And it's a big reminder for us all that behind every clinical trial, it's not just data, it's actual patients, their experiences, their strength, and their trust in us. And I think it's a big reminder for why we are doing these trials and why doing research is important.
yeah, I really appreciate it, because even questions, I try to be really good about writing stuff down and asking when I'm there, but the my chart is amazing, and I'm not really tech savvy, but I am able to get on there, and questions are answered right away. I get a quick response, and even if I don't get an answer, I at least get a response that says, "Don't know the answer, I'll get back to you." And yeah, and then I get my questions answered. So that's nice. It's nice not to feel like just a number. So that's I appreciate that.
Of course, absolutely. It's our privilege to take care of you. Do you have any advice for maybe a patient who's considering enrolling in a trial or might have some hesitance, anything that would help make that decision a little bit easier for them?
I mean, I just say, ask questions. It's been nice because at one point I did have, you know, I shouldn't say bounced around, but I had another doctor and everything was like, are you sure you're okay with this? Did you want to go back to your other doctor? Do you have questions for your other doctor? Um, you know, so the teamwork is very nice that everybody is talking to each other. And again, I don't feel like I'm being pawned off. I think that it's nice that you know, you should get what you ask for and not just be a pawn. If you don't like your doctor, then ask for a different one. But, you know, get your questions answered and feel comfortable, because that, I believe, is part of the healing process, is that you have to have a team that you feel confident in. If you don't, then you're not going to go anywhere.
Definitely, that's a very, very important aspect. Thanks so much for sharing all of that with us, and thanks so much for joining.
You're very welcome. Thank you.
Well, I guess thanks everybody. Thanks Afsaneh for putting this on, and thanks Dr. Tao, Dr. Lee, and Dr. Tsai. And hopefully everybody has a great holiday and has some nice plans for Thanksgiving too. So with that, I'm going to end. And anybody have any final thoughts?
Just wanted to give a quick shout out to our amazing admin and clinical admin team, especially Jen, who helped out a lot with some of our last minute troubleshooting, and for Veronica, Cole, they've been all super helpful in spreading the word about this conference, and I appreciate their help organizing this event.
Great. Thank you.
Thank you so much, everyone.
Yeah,